Hello, welcome to my DNA replication model. As you can see, we have the nitrogenic bases, shown adenine being green, thymine blue, guanine orange, and cytosine pink. The base pairings are seen as adenine and thymine, and guanine and cytosine. Adenine and guanine are purines, and cytosine and thymine are pyrimidines. Purines have 2 rings, and pyrimidines have 1 ring. You can remember purines have 2 rings because it sounds like pure, meaning it is complete and intact, while the pyramids have sharp edges, so pyramids have had their second ring cut off. Then we have the hydrogen bonds between the nitrogenous bases. Between cytosine and guanine, there are 3 hydrogen bonds, and only 2 between adenine and thymine. The nitrogenic bases attach to the sugar in the backbone of the DNA, and between each sugar is a phosphate. The replication bubbles are used to increase speed of the process of DNA replication, as there are multiple on a strand of DNA at a time, and they are formed by the separation of 2 DNA strands by the helicase. The helicase is an enzyme that separates the strands of DNA, forming the replication fork, which is called that because of the forked appearance of the strands of DNA. The helicase works similarly to a zipper. By unzipping the 2 strands of DNA, this is because the incoming triphosphates need to be able to form base pairs. The topoisomerase works hand in hand with the helicase by stopping the overwinding or supercoiling of DNA strands when they are unzipped. Without the topoisomerase, the DNA would break during separation. The single-strand binding proteins hold open the 2 DNA strands so they don't reform a double helix after the helicase comes through. Then there are the DNA polymerase III, which adds the nucleotides to the primers. The leading strand is synthesized in one piece, needing only an initial RNA primer to begin synthesis. RNA primer is a binding site for the DNA polymerase, and DNA polymerase is only able to add nucleotides to an existing nucleotide chain. Therefore, RNA primer needs to start that chain. Nucleoside triphosphates bring in 3 phosphates. When the bond between the nucleotides and phosphates is broken, energy is released, which is used to bond the nucleotides to the growing chain, which creates a growing strand. DNA polymerase I has more function in the lagging strand than leading strand, as its purpose is to remove RNA primers in the processing of DNA after replication. In the lagging strand, there are Okazaki fragments, which allow for the daughter strand to be synthesized. The RNA primer on the lagging strand is more common as DNA is created in small lengths. Each of these fragments must begin with RNA primer. The DNA ligase is used to bind the Okazaki fragments together. DNA polymerase I in the lagging strand removes the RNA primers and fills the gaps by synthesizing DNA. DNA primus catalyzes the synthesis of short RNA molecules used as primers for DNA polymerase, which basically just means they synthesize of the discontinuously synthesized Okazaki fragments. The reason the lagging strand is discontinuous and creates the Okazaki fragments is because the lagging strand must wait for the leading strand to expose the template strand, which is the strand that is used during transcription to provide RNA, and the template strand is on which the Okazaki fragments are synthesized. That's all for my DNA replication model. Have a nice day. Bye!